Unfortunately, I didn't record lecture capture. I didn't have the thing on. So I'm going to start here. I'm going to talk about basically neurobiology. But first I want to mention that organisms have to respond and interact with their environment. Not the only group that's evolved so far that actually has a nervous system that actually led to perhaps intelligent organisms is the animal kingdom. But all these other things actually interact as well. Bacteria most often interacts with chemotaxis, which is chemicals. In fact, chemicals are used by a wide variety of organisms to interact. A lot of insects use chemicals. Mammals use it. Tons. Plants do something called phototaxis, where they actually move in relation to the sun, and that photo is sunlight or light. Plants also do other things. If a plant, let's say a Jeffrey pine, a pine tree is being attacked by bark beetles, they respond, of course, with a kind of a simple immune system, but they also release these volatile chemicals that other nearby trees pick up on, that alerts them to the fact that, oh, this one tree is being attacked by bark beetles. And they immediately mount their own immune defenses, so it's already ready before the beetles actually show up. Now, nervous system, it's unique to the animal kingdom, as I mentioned. Most animals have nervous systems of some sort. The only animals that don't are sponges, but everything on up, like sea anemones, jellyfish, let's see, worms, all sorts of different things have nervous tissue. The simplest is like a neural net. There's no real brain, but they can perceive things around them. And then, of course, with us, all our sense organs are kind of tied into our nervous system, right? So that's how we kind of engage the world around us. So reflexes doesn't require thinking, right? It's just basically a very simple sensory neuron, let's say a hot stimulus, so this is touch. An interneuron within the spinal cord that carries the signal. A motor neuron that then initiates the response, which is, in this case, withdrawing the hand from the hot stimulus. And then there's an effector. So the motor neurons, and then the effector is actually the muscle or tissue that's stimulated by the motor neuron, the charge that comes down it, and causing individuals to actually do something. So a reflex is an involuntary movement of a body in response to a stimulus. A knee tap, reaction to unexpected painful stimuli. Salivation in response to the smell of good food. Gag response exposed to noxious stimuli. Reaction does not involve cognitive processing. And there's a reason for that. Let's say a monster came in the room, and we looked up, and what would you see? What would you see? And immediately you'd have a startled response, and you'd do the fight or flight response. It doesn't take any thinking to do this. In fact, you don't want to think, oh, there's a monster in the room. What should I do? Because by that time, everybody else is going to have run away, and you're the one left over to be eaten by the monster. So, neurons direct basically behavior. You have sensory neurons, interneurons, motor neurons, and the effectors. So the nervous system can be divided into two parts. The central nervous system, CNS, is the brain and spinal cord. The CNS is well protected in humans. We have a vertebral column made of bone, and then we have a skull which is strongly reinforced with different layers of bone. And then beyond that, we have a peripheral nervous system. In other words, if along the spinal column between each disc, there's going to be neurons coming out, branched neurons, that then will go to all the different muscles and everything else in the body, all the individual cells, and that's how responses are made. So the peripheral nervous system is the tail end of everything. But it's very important because the peripheral nervous system also reacts to incoming stimuli. So here's the central nervous system. As I mentioned, it's strongly protected. You can see how strongly the nervous system is protected, right? And then the spinal cord is right in the middle, heavily protected. And then here's these side branching neurons that come out. In the upper part, the thoracic vertebrae, it goes to your arms and neck, and then the lumbar, thoracic, central of your body, and then lumbar, lower body. For example, to the legs. The meninges kind of protects the brain. Let's take a look at this more closely. Okay, so the meninges and the meningeal layer is a membrane that protects the brain, and then inside it is a spinal fluid, protects the brain and the spinal cord. Now, let's take a look at this. And then, of course, there's something we'll talk about a little bit more, but there's a blood-brain barrier that actually restricts things from entering the brain to protect the brain. So, for example, the only kind of fuel that the brain can actually use is I think it's glycogen. This blood-brain barrier it's a great thing, of course, protects us, but it's also, in terms of drug delivery to the brain, it makes it much more difficult. So, here we are, the blood-brain barrier. Now, capillaries are the smallest blood vessels, and in the brain, they're almost impermeable. In other words, they stop almost everything from getting through. Okay, let's look at this. The cerebrum controls language, memory, sensations, decision-making, executive functions. The skull, of course, the cerebellum is responsible for balance and muscle movement. The brain stem is part of this autonomous nervous system. You don't have to think about your heart beating or breathing or swallowing. You just do them, right? And then if that's damaged, they'll put someone like life support, but they're pretty much gone. Thalamus relays sensory information, kind of in this area, we call it kind of the limbic system, our reptilian brain, where we kind of react to things like reflex actions and things like that. So, brain death, no signs of brain activity at all. A recording of brain activity is called an electroencephalogram. A coma, there's some signs of brain activity. If there's no brainstem damage, then you don't need a respirator. Comas vary in how severe they are. In some cases, it was commonly thought that people who were in comas actually they weren't aware of their surroundings. But that changed, I guess about 15, 20 years ago. there's a guy named, a neurologist named Dr. Sachs. And there was a movie about him. Robin Williams was in it about, he was on a ward, and there were all these people who were in a coma because there was an outbreak of St. Louis encephalitis, which is kind of a sleeping sickness. And they all kind of lapsed into a coma, and they'd been that way for many, many, many years. And then he decided to, they were learning more about stuff, and he decided to inject them with dopamine, and it turns out that woke them up. And this worked for a while. After a while, it didn't. They went back. But in the meantime, all these patients mentioned that they were in this kind of, half-conscious state. They could actually think. They could hear things around them. They just couldn't respond. They were basically captive in their own body. So neurons are obviously highly specialized cells. They don't normally divide. So damage, for the most part, to neural tissue is permanent. Though we're trying to change that with like stem cells and, let's say, spinal cord injuries. And there's been very limited success. There was one guy, they kind of put the scaffolding, and they grew stem cells between the two sides of the spinal cord, and there was a thin layer of neurons that then filled in, and he had very limited movement. So the neurons basically have kind of three parts. Now, the dendrites collect signals. They're not exactly electrical signals. They're chemical electrical signals. We have the cell body that contains the nucleus and organelles that you are familiar with. Here's the axon. Delivers these signals to dendrites of another cell by going finally to the terminal, where neurotransmitters, these kind of hormonal chemicals, are released. So for a nerve impulse to actually happen, you need an entry, a nerve impulse. So we have something action potential. It has to be at a certain level or it doesn't get transmitted. You have ions. Now ions are positively or negatively charged elements. And they carry the electrical charge down the nerve cell. So here we are. Na is I think it's sodium. K is potassium. And then there's usually negative charged particles as well. And so this kind of potential in terms of positive to positive to positive kind of carries the signal down. It's all chemical ions, though. It's not like a real electrical signal. Now, neurons are protected. They actually have a myelin sheath. It's this thin covering of myelin cells. They're called Schwann cells, actually. And they protect the neuron, and they also actually allow the impulse to carry down the neuron without leaking out. So, now gray matter is unmyelinated axons, but white matter is the kind that is myelinated axons. And without it, you get leakage and less conductance. This is, people with multiple sclerosis have a problem. Their immune system destroys the myelin sheath, and they have degenerative, neurodegenerative diseases. here we are with multiple sclerosis. And so immune response against myelin, this is all broken down. And unfortunately, they become less and less, the neurons are less good at carrying these different action potential signals. So there's some recent advances in MS treatment. They have this antigen-specific therapy. This distracts the rogue immune cells. So you put these antigens specifically, and instead of the immune system attacking the myelin, it actually gloms onto the antigen and doesn't have a chance to do the damage. There's also some new small molecule therapy. I guess it's these are pretty expensive now, but the first time that actually MS has been treatable. And then there's this thing about since it's an autoimmune disease, it turns out a lot of autoimmune diseases have their basis in the immune system not being properly trained and not having the right gut bacteria. And so I've heard anecdotally that like there was a guy, I guess a physician in Australia who took a fecal transplant from one person, put it into another. Those bacteria and everything that was in it basically kind of took over. The patient who had MS didn't have a very good, gut bacteria and stuff, and then that actually reversed some of the effects. So there's a number of things they've done. Also, stem cell therapy, of course, replaces of defective immune cells. In other words, cells, instead of the cells that were attacking the body, you replace them with cells that don't attack the body. And then replacement of lost myelin producing cells. Okay, now we're getting to neurons and neuron function and how it works. So we mentioned here we have an impulse. These are neurotransmitters that come across attached to receptors in the dendrite. These ion signals are then carried down the axon to the axon terminal. And then, if we actually look at it a little more closely, right in between is the synapse. In other words, the terminal and the next dendrite for the next neuron, there's a space in between. It's very narrow, but they don't touch. Okay. Okay, here we go. So, signals are actually carried across by chemical neurotransmitters. So we call it an electrical signal. It's ions, but it's positively or negatively charged particles. Okay, and these neurotransmitters are then the ones that carry the signal across from the terminal branch, right here, to the new dendrite. Okay, so let's see. Here we have presynaptic neuron transmission of an impulse across the synapse. Terminal. This is the terminal end of the neuron, and this is then the dendrite end. And then we have these neurotransmitters that come in vesicles. Okay? And as they come out, they're released. And they actually move across the synapse, and they hit receptors on the other side that uptake those vesicles. I mean those enzymes, whether it's serotonin or dopamine, various other kinds. And once they all fill up, if there's any left over, we have them digested by enzymes. At the same time, there's also those that haven't been taken up by, in this case down here, are then reuptake and back into the dendron, into the neuron terminal axis, into these vesicles. Some examples of neurotransmitters that interact: acetylcholine, dopamine, serotonin, glutamate, GABA, the norepinephrine system, whole bunch. They're all different. Neurons use different ones. Many neurological diseases are really linked to problems with different neurotransmitters. In some cases, too many neurotransmitters are being released or not enough. In this way, you can actually treat some of these, let's say, diseases by affecting the specific levels of neurotransmitters. So if there's not enough, you can perhaps get cells to generate more. Or if too many, and you want to stop the uptake for the next signal, you put in something that gets to the little area where the neurotransmitters block, and it blocks the uptake. So one common disease that neurotransmitter imbalance is partly responsible for is depression. Usually because of low serotonin levels. And so what you do is you actually, there's a drug that acts to increase serotonin levels at the synapses. And let's see, Zoloft, Prozac, Paxil, Fexor, there's a whole bunch of new ones. They're more specific than what was used in the past, and therefore more effective, but still not as good as they could be. So here's serotonin again. So what happens is SSRI is a selective serotonin reuptake inhibitor, and it works by preventing serotonin from being recycled. It stays in the synapse longer, and it continues to propagate the impulse. Most drugs that affect the nervous system do so because they affect the levels of specific neurotransmitters. So drugs that affect serotonin are SSRIs, so serotonin selective reuptake inhibitors. Drugs that inhibit reuptake allow the neurotransmitter to remain in the synapse longer, right, and do their thing. Now, it turns out that a lot of these receptors here and here, that they not only do these receptors uptake, the neurotransmitters, but they'll oftentimes drugs will react with them and so let's say take their place. So let's see. For example, let's take a look at this. Let's say we have LSD, and it gets in here. LSD actually let's see. Oh, there's some all the way here. Okay. But let's say LSD will come in and get in the way. So that's how it actually works. MMDA is the same thing. Just so you know, LSD is not addictive because once all the it works on the dopamine system, by the way. Actually, it doesn't. I don't think it actually gets in the way. I forget. But what it does is all these neurotransmitters are released, and there's a low amount then are left over, and in terms of dopamine, and so usually you have to wait like 3 or 4 days after taking LSD for it to have the same effect again. So it turns out these drugs can mimic the neurotransmitter itself, activating receptors on the postsynaptic cell. That's right, I was right. Okay. So, MMDA, ecstasy, part of the same group, though it works on the serotonin pathway. Feelings of well-being, also interferes with thermoregulation. Depletion of serotonin stores, slow to resynthesize. So usually you take ecstasy, if you take it the next day, it's not going to have much effect. Attention deficit disorder and dopamine. So low levels of dopamine, possible due to excess reuptake of the transporters, and Ritalin actually blocks the reuptake, so you have more staying between the synapse and actually working. Just so you know, okay, Ritalin. Let me just mention one thing, though. There seems like an increase in, let's say, attention deficit disorder, all sorts of cognitive problems in kids. Now, why is this happening? Well, perhaps there are environmental toxins that are perhaps, causing it. There's a hypothesis. The thing is, there's variation in the levels of neurotransmitters in different people, and you see the results. So some people are it's hard for them to pay attention. They're edgy. They want to go do something else. They can't kind of focus on one thing. Now you think, oh well, that's not good, but they also tend to go out and explore things. So think about it before the modern world. We were all hunter-gatherers. Some people were good. They'd stay at home. They do what they're told. Da-da-da-da. Others, kids would go out and run around. Some would get eaten by saber-toothed tigers. But some also, they were exploring, finding new things. And that was adaptive. So they might leave more offspring. So this attention deficit disorder and things like that may just be a function of naturally occurring variation, and that behavior in the past might have been favored. It kind of gets crushed in schools. Doesn't mean that the people who are, ADD are bad people. They just can't keep their focus. Oftentimes they get through it. Things stabilize a little bit, but they're still always kind of searching and finding new things. And if they can get past the bad part, they oftentimes do very, very well. So in terms of Ritalin, it inhibits dopamine reuptake. So let's see. So normally reuptake of dopamine limits signaling. Here's a dopamine receptor. These are the dopamine synapse. And then the Ritalin is this. And what it does is it stops the reuptake of dopamine, and it regulates attentiveness. Now the thing is, Ritalin works in a similar way to cocaine. Ritalin, kind of a low dose, slow release, hopefully a decent effect. Maybe don't need it at all. Cocaine, high dose, immediate effect. It kind of enlivens the pleasure centers of our brain. Now, Ritalin, these both are drugs. They both affect the receptors. How is it that we have receptors that are similar to drugs that are similar to neurotransmitters? Like we have opioid receptors, heroin, morphine, synthetics like oxycodone, hydrocodone, all that kind of stuff. That's how a lot of these mind-altering drugs actually work. Now, as you know, probably some drugs are addicting and others are not. I mentioned LSD and MMDA. Neither of those are addicting because you deplete the neurotransmitters and it takes a while for them to be synthesized, manufactured again. So it might take, let's say, 4 days to get the same rush from LSD that you had before. One day it's going to be much diminished. So that's not going to work. But many addictive drugs, there's an immediate effect. That could be LSD. Euphoric, same thing. But and it works kind of a short duration. But like I said, it doesn't exactly work. Now, the thing is, if it activates the pleasure centers of the brain, and there are many things that do this, so not just the drugs, but like gambling, that same sort of thing, that same pathway. But if the drug is capable of inducing a tolerance, in other words, an addictive behavior, then it can become addictive. Now, I'll give you an example of tolerance. If you take someone who's, let's say, a heroin addict, and they take, let's say, a certain dose of heroin to get a certain high. If you gave that dose to someone who wasn't a heroin addict, it's very likely that you would give them an overdose. Because when someone first starts using, they're very sensitive, in this case, to the drug. But over time, they build up more and more tolerance. And so you have to take a larger and larger amount of the drug to get the same effect. Here's another one, methamphetamine, highly addictive. So it decreases number of dopamine receptors, so you need more and more stimulation. This is known as tolerance. Just so you know, meth addicts, it's probably the worst thing you could probably get addicted to. Okay, now this is what methamphetamine does. People start to melt. You probably think, what is he talking about? Well, if you've seen a meth addict who's been at it for a long time, they start getting these lesions on their skin, and their skin kind of, they look, within a few years, they look like they've aged 20. And inside, it's just as bad inside their bodies. They also tend to be paranoid. A lot of things go along with it. If you're going to get addicted to something, don't get addicted to methamphetamine. Besides, it's really, the only people who use it are, like poor white trash. Okay. We'll stop there, and we'll continue next time in class.